Okay, so good evening. I'm Kishan here, and I'm going to teach you today the M-phase regulation of cell cycle. Okay. So before start, I would like to know whether my voice is clear and you are able to see this particular screen, which is showing events of M-phase and stages of mitosis. Anyone, please tell me if my voice is clear and you are able to see the screen. Okay, thank you, Krishna and Ankita. Okay, fine. Everything is fine, and we can start the class now. So, in today's class, we will discuss what we have left, where we have left actually in our last class. So, in our last class, actually, we have left over this particular events of M phase. So, actually, during M phase, because this is the most dramatic period of cell cycle, major things happen in this particular phase, and these include a nuclear division, means the chromosomes need to condense to make, means these chromosomes are formed out of chromatin. What is the basic difference between chromatin and chromosome? You should able to understand this during S phase of cell cycle when DNA is replicated, and this DNA is long thread of means polynucleotides, but this particular length of DNA should be reduced to make compact chromosomes. See here. So here, for example, here. Okay, so fine. So the cell, when the cell is in interphase and it continued to grow, you can see the size difference here and here. The complete cell growth has been taken place, and because interphase also include the S phase, so during S phase DNA replication has been taking place, so that the size of nucleus is also increased. But when the cell move through G2 to M phase, and the M phase include the prophase, metaphase, anaphase, and telophase, during that phases you can see clear chromosomes, means this particular chromatin material which is dispersed throughout the nucleus got condensed to form the proper chromosomes. So therefore chromosomes are nothing but the condensed form of chromatin. Okay, so you can get it. One another diagram I showed you yesterday. Okay, so here, okay, that is fine. So that's how this length of chromosome, this DNA, both chromatin and chromosome are DNA, but the length of this particular polymer is reduced by several compactation happening actually with the help of some proteins. Some of them are called cohesins, which are present between two sister chromatids, and these cohesins from the sister chromatids are removed and replaced by the second class of proteins called condensins, which help in condensation of chromosomes or to reduce their size. Okay, so you can think that in the terms of, जैसे आप लोग कभी पतंग उड़ाते हैं तो जो धागा होता है ना, तो जब पतंग को दूर छोड़ना होता है, दूर भेजना होता है, तो हम क्या करते हैं, धागे को रिलीज करते हैं। तो और उसके बाद जब पतंग को वापस से खींचते हैं तो क्या होता है कि वो धागा जिससे वो टेंगल्ड ना हो, उलझे नहीं, सो उसको क्या करते हैं, साथ साथ लपेटते जाते हैं। तो सिमिलरली, because during nuclear division or chromosomal segregation or DNA segregation between daughter cells, there is a possibility that when these sister chromatids are pulled off towards the opposite poles, they can break from in between. So अगर DNA का breakage हो जाएगा, DNA टूट जाएगा, then it will be harmful to that particular cell type. So therefore, they are compacted. They are means the compact forms of chromosomes. Okay, so that it is easy to segregate them during the nuclear divisions or karyokinesis. Now we came back. So in today's class, actually, we will discuss from here. This we have already discussed about cell cycle. So cell cycle is sequence of several phases, including G1, S, G2, and M phase. These three phases are called interphase, and the cell ultimately reaches into a phase called mitosis phase or M phase, where karyokinesis is followed by cytokinesis. Now you have seen this particular diagram also. The central portion is called centrosome, And actually, the cohesin and condensin proteins help in compactation of chromosomes, so that these kinetochores are able to attach to the mitotic spindle. Okay, so now what is the requirement here? When these chromosomes are arranged over metaphase plate, so this particular protein that is called cohesin, cohesin is remaining only at this particular portion, that is means in between the two chromosome. The sister chromatid are joined by these green colored proteins called cohesins. So these cohesins should be broken down so that the two sister chromatids can be segregated or separated. How this is happening within the cell? Okay, so first of all, there is two proteins are making an inactive complex. One protein is called securin, another protein is called saprase. सिमिलरली जैसे सीडी के और साइक्लिन कॉम्प्लेक्स बनाते हैं, वैसे ही ये दोनों प्रोटीन भी इनमें से एक इनहिबिटरी प्रोटीन है, या हम कह सकते हैं इट इज हैविंग रेगुलेटरी फंक्शन. और जो दूसरी प्रोटीन है सैपरेज, इट इज हैविंग सम कैटालिटिक फंक्शन एंड इट इज अ सिस्टीन प्रोटीज. I want you to note this particular point, that this is a cysteine protease. It is also written in your Pathfinder book also. Okay, so if you move from here to here, wait a minute. Okay, so there is two proteins making a complex called securin and saprase. If they are together, they are inactive, but they are activated by certain proteins called APC, anaphase promoting complex. Okay, so when this is bound to CDC20, okay, so they are able to degrade the securin, which is having some regulatory role or inhibitory role over this saprase. When saprase is active, it is able to break down the cohesin protein between the two sister chromatids, and thus the two sister chromatids can be segregated out. Okay, so from the text you will find out this, that the securin hold an enzyme saprase. This saprase is a ubiquitous cysteine protease. Underline this line, I like, because this is very important, that this is a cysteine protease. It will specifically cleave at cysteine amino acid in your target protein. That target protein is cohesin. Okay, now we came back to this particular diagram. Okay, so this is your cohesin here, and this particular cohesin is broken down by a protein called saprase. So saprase separates the two sister chromatids by breaking down this particular protein. Now, okay, so that the chromosome which are aligned over metaphasic plate can be segregated, and this metaphase to anaphase transition can happen. Okay, so now you may have a question that how this molecule, how this cohesins are able to hold two sister chromatids together, and how they will be broken down. Means if you see here, these are two sister chromatids. This is one, this is second, and the green color protein, that is cohesin, is encircling or means binding your two sister chromatids here. So how this is happening, you will find it from this particular diagram. See, this cohesin protein is a dimer, means this particular dimer is having C to N terminus, N to C terminus. So at one end there is a C terminus, there is N terminus. This is called SMC molecule, or structural maintenance of chromosome molecule. This is a dimer, and at one end where its N terminus and C terminus are together, it is having a ATPase domain from where it can hydrolyze the ATP. Okay, so when this particular monomer is attached to second means dimer, attached to second dimer, so it is forming a hinge region together, and the second protein is actually the isomer of this particular protein, structural maintenance of chromosome, structural maintenance of chromosome third, and this is one. So these are making another ring-like structure, and both the ATPase domains came together, which are in turn interacting with another proteins, which is called SCC1, and this is called SRC or SCC3. Okay, so actually not very visible from this particular PDF, but I'll try to like this. So this is SCC3 and this is SCC1. So this is your chromatin. Okay, so the two sister chromatids, the DNA at that particular portion is wrapped by this particular polymer of SMC3 and SMC1, means cohesin. So that's how at hinge region, okay, so this protein or the cohesin protein, this complete protein is cohesin, which is encircling your sister chromatids. Okay, so it is holding both the sister chromatids together. So it must be broken down to release the two sister chromatids so that the sister chromatids can be separated during anaphase of your M phase. So see how this is happening actually. So because the Securin holds this particular enzyme called saprase, and this is a cysteine protease in an inactive state. If somehow you are able to destroy the Securin, you will free the saprase enzyme, and this active saprase enzyme is able to remove this cohesin proteins from this particular portion so that the two sister chromatids can be segregated, can be separated or segregated. So the destruction of Securin allows saprase to cleave the SCC1, a subunit of the cohesin which hold the sister chromatids together. So the target is SCC1. Now see again, this SCC1 is this blue color protein, and SCC3 is only a portion of this. So the major protein which is holding the ATPase domain together is SCC1. If this is broken down, your ring will open and the two sister chromatids will be segregated. Got this? Okay, if you have some query from this portion before moving to condensin, which is helping the chromosome to condense, the major proteins which are holding the sister chromatids together are these cohesins. And I have told you that the saprase enzyme is responsible for breakdown of this particular protein SCC1. And once the SCC1 is broken down, this particular ring will open and the two sister chromatids will be separated, means the chromosome will be free to move. For example, in this diagram, once this is broken, the two sister chromatids will be moving towards the opposite poles in the anaphase. So the cell will move from metaphase to anaphase. This is called anaphase promoting complex protein, and this anaphase promoting complex protein is helping in this particular ubiquitinization and degradation of Securin protein, freeing the saprase enzyme, and therefore the saprase is able to degrade the cohesin proteins so that okay, so this type of breakdown is not direct, but indirectly it is breaking down the Securin, not directly breaking down your this cohesin protein, but indirectly it is moving, it is helping cell to move out of metaphase so that the cell can enter into the anaphase, and thus the cell cycle will continue. If someone have query, please ask, or I will move to the next protein. I'm not clear with the separation. How does separation happen with the SCC1 opens ring? Trishla, see again. I hope it is okay. I will explain again. There is no problem because it is my responsibility so that I am able to clear all the topics what I'm teaching you. So see here, this Securin holds an enzyme called saprase. So there is two proteins, one is called Securin, one is called saprase. If it is not clear from here, I will also have the other thing here. Sorry. Okay, so there is two proteins. Again, I'm starting from here. So there is two proteins. One is having some regulatory function or inhibitory function that is called securin. And another protein, which is an enzyme or cysteine protease, is called saprase. Now see, this particular enzyme or protein is inhibiting this particular protein, this particular enzyme. Okay, will not allow it to act over, over this cohesin, which is holding your sister chromatids. So till here, you have studied four proteins, different proteins: cohesins, condensins, securin, and saprase. Cohesin and condensins are required for the compactation of chromosome, uh, for the formation of chromosomes, proper structures, whereas securin and saprase are required to separate the sister chromatids to degrade this particular green protein, which is present in between these two. Actually, what is this green portion? This green portion is shown in the next diagram. This green portion is actually this complete protein ring-like structure around this DNA molecule. So these are DNA molecules wrapped over histone proteins. So okay, so this is the portion of portion where sister chromatids join together in a chromosome with the help of this particular ring. So if your protein that is called saprase is freed from this particular protein called securin, so how it will be freed? So first you need to degrade this particular protein. You need to remove this. So I will like to, so I'm using some annotations. So you need to remove this particular protein that is called securin, so that this particular protein saprase become activated. Once it will be activated, this is a cysteine protease, and so to remove this, you need to do the ubiquitination, so that the proteasomal degradation will result in the degradation of this particular protein, securin. Once this is degraded, here it is degraded, and this particular enzyme called saprase will be activated. You can see these things are coming out of this enzyme, means the conformation is changed when it is freed from securin. So now it is activated. So once it is activated, so it will remove all these ring-like structures which are holding the sister chromatids together. Actually, they are showing too many rings in this particular diagram, but when the chromosomes are compact, these rings are generally present in this portion where they are holding two sister chromatids together. Okay, so this cohesin complexes should be broken down with the help of this saprase enzyme or protein. Once they are broken down, the two sister chromatids will be separated and they will start moving towards the opposite poles. Okay. So sorry, actually I have annotated this and it will move everywhere. Fine. So actually this is the proper structure of your cohesin. It is having SMC molecule that is called structural maintenance of chromosome 1 and 3. So this 1 and 3 is forming a ring, and this SCC protein is acting as a seal over this ring. So this seal should be broken down by the saprase enzyme so that this ring will open. And once this ring is open, do not look into the another diagram. This is different. I will describe it in the next. Okay. So once this is open, the seal is broken and this ring is open, the two sister chromatids will be segregated. Two DNA molecules will be segregated. So hope you will be able to get this. And if you are not clear, oh, thank you. Here, sir, I want to know SEC and SMC protein have to make the cohesin condensin protein or not. SCC and SMC, yes, they make the cohesin and condensin proteins. Yes, beta, I'm coming to the second portion that is called condensins. Up to here, I'm actually describing the cohesin proteins. The condensins are also having the similar structure. I'm coming to them. Okay, so I will come soon. I'm not clear with the separation. How does the separation happen when the SEC opens the ring? The SEC is not opening the ring. Actually, once the SEC is broken down, because it is acting as a seal, once this is broken down, it will open up by itself. Okay. So because it is holding both the polymers of SMC1 and SMC3, sorry. So once it is broken down, they will open up. There is no requirement of anything. Now we are coming to another protein which are required for the condensation of chromosome, that's condensins actually. So here you have seen there is 4 monomers. One is called SMC1, SMC3, SCC1 and SCC3. Here in condensin there are 5 proteins which are making the complete structure. So we'll see. Now we are moving to condensins. So, okay, so you will get the difference. If you want to compare, I am putting it here. So here it is SMC molecule. Here also there is SMC molecule, but here the SMC molecule will 1 and 3. Here the another homologue or isomer of this SMC, that is called SMC2 and SMC4 were present. So remember, SMC1 and SMC3 are making your cohesins. SMC2 and 4 are making your condensins. Okay, Shreya? Okay, fine. So it is also having an ATPase domain, and this ATPase domain is bound by 3 proteins, and this seal is made up of cap H, and the cap H and the 2 sides are also joined by cap G and D2. Now there is 5 proteins which are making this particular ring, and this ring is holding your DNA, compacting your chromosomes many times. Like this is the simple structure, and when there is long runs of DNA, they are making another ring like structures by arranging their head-like structures in the ring form, so that the chromosomes are condensed like this, from this to this to this. So actually you will be surprised to know that this particular protein condensin is able to condense your DNA to make a proper chromosome, and this condensation is about 1000-fold. Means from the simple DNA that is called chromatin to make chromosome, DNA is compacted about 1000 times. Okay. So to make this particular complex structure where these structures or ring-like structures are in continuation. So now I will lead you to the simple diagram. Okay, so here it is given in the form of dots, but these dots are not so simple. These dots of condensins are something like these dots. So they are holding your DNA to make compact chromosomes. Hope you are clear, because very simple presentation, very simple things you are able to see from here. And if you are okay, sure. Okay. Any queries, any doubts? You may ask. Now we are moving to our next discussion. Okay, yesterday someone has asked me one question, Krishla, I think. What is the mechanism? What is the molecular mechanism? How CDK1 enhance the cyclin B degradation? How this cyclin B is degraded? How other proteins which are regulating the cell cycle are being degraded? And I simply told you that actually this all happens through ubiquitin mediated proteasomal degradation. So in today's class, I will describe what is proteasome, how this ubiquitin mediated proteasome degradation will taking or happening within your cells, and what are the components which are required for this to happen actually. Okay, so there are some queries. 1 minute. Okay, Shreya, actually these proteins are having similar function which are acting as a seal over your particular condensin protein, but unlike Okay, so from the diagram only, I will like to describe it, okay, so that you can get the point. So, see, this is cohesin and this is condensin. This cohesin protein is holding your sister chromatids and this condensin is holding your DNA so that the DNA can be compacted to form chromosomes. Beta, to separate sister chromatids, you need not break condensins. You only need to break this particular polymer or this particular protein of cohesin. Okay, so that the sister chromatids can be separated from each other and can move towards the opposite poles. There is no need to break down this, but actually this is having similar function as these two proteins, SEC1 and SEC3, are making a connection between these two SMC molecules, structural maintenance of chromosome molecules. Similarly, this cap H and cap D2G all are making so that these two SMC molecule 2 and 4 can be held together so that all the DNA can be compacted. These need to be broken down when interphase will come again. Okay, so during interphase, this seal will be removed, and okay, that is not part of our discussion. In this class, we need to study only cohesin degradation, okay, so that your cohesin will be degraded, and once your cohesin complexes will be degraded, your chromosome will move towards the opposite pole, so that cell can move from one phase into the another, means from metaphase to anaphase of your M phase of cell cycle. Fine. Okay, so now we will move to many things we have discussed in yesterday's class. We have discussed CDK activating kinase. Yes. We have discussed V1 kinase, which is actually this is a kinase, this is a phosphatase. We have seen that they are having opposite functions. CDC25, oh sorry, both are kinases, but they are having different functions. Means this CAK phosphorylate your threonine at 161 position, which is having some activating effects, whereas V1 kinase, sorry for that, this is not a phosphatase, this is a kinase. I'm repeating again, this is a kinase, but it will phosphorylate your inhibitory position, means generally your tyrosine 15. Okay, so phosphorylate inhibitory sites of CDK, primarily involves suppressing CDK1, so that it can suppress this particular kinase activity, CDK1 activity in MPF, maturation promoting factor. Then this is a phosphatase, CDC25, and this and actually this kinase V1 and this 25 phosphatase are having opposite function, because this is phosphorylating your inhibitory positions, inhibitory threonine or tyrosine positions, whereas CDC25 is removing all the phosphates from those particular positions which are inhibiting the CDK1. And there is three members in this family, A, B, C are found in mammals. We have seen A and C yesterday. Okay. Then there is certain inhibitory molecules that is known as one, that is SIC1, which is present in budding yeast. P27 we have discussed, which is inhibiting, okay. So during your S phase of cell cycle, this P27 we have discussed in retinoblastoma portion. P21 we have discussed in P53 portion. We have discussed day after, means परसों जो क्लास हुई थी उसमें हमने डिस्कस करा था. Okay, similarly P16 is also having some inhibitory roles during G1, suppressing the G1 CDK activity. This G1 CDK activity is to suppress your cyclin D, which is making complex with either CDK4 or 6. Okay, and this P16 is a member of INC4 family. I have told you in yesterday's class. Now, beside this, because we have discussed majority of them, now in today's class, actually my topic of interest is to discuss the ubiquitin ligases and their activators. Because these ubiquitin ligases are family of enzymes which are required for ubiquitination. What is ubiquitin? Ubiquitin is a protein which is used to tag those particular protein targets which are अंडरगोइंग प्रोटियोलाइसिस, यानी कि जिनका डिग्रेडेशन होना है, उन प्रोटीन्स को डिग्रेड करने के लिए उनके ऊपर यूबिक्यूटिन का एक टैग लगा देते हैं, सो दैट प्रोटियोसोम कैन रिकॉग्नाइज those particular proteins and can degrade them by using ATP. नाउ जो इस क्लास में आते हैं वो हमारे एनाफेज प्रोमोटिंग कॉम्प्लेक्स APC, ओके, CDC20, CDH1, CDC20, homolog 1, okay, and SCF. What is SCF? So we will see SCF also, because somewhere it will come. So actually we need to discuss all these things in today's class only. So you will find where it was. So the full forms are given in your book. Wait a minute, I missed. Okay, it is here only. So when you go back. So when you start this particular topic, you will find SCF. Okay, so this particular enzyme SCF, which is made up of 3 different proteins, you can underline it from here. SCF is a combination of SKP1 protein, Cullin 1, COLLIN, and F box protein. I will show you the detailed structure of SCF and how it works in molecular mechanism and all these things we will discuss in this class only. So before starting that portion, I would like to tell you some important things from this text. So cyclin undergoes cycle of synthesis and degradation in each cell cycle. We have discussed this thing because cyclins are regulatory portion of our cell cycle, and these are required to make complexes with some of the CDKs, and this CDKs are cyclin dependent kinases. Okay, so CDK levels will not change, only the synthesis or degradation of cyclins will happen or take place during different phases of cell cycle. So cyclin changes in the cyclin levels result in cyclic assembly and activation of the cyclin CDK complexes, inactivated by regulated proteolysis of cyclin at specific cell cycle stages. तो जब भी cell cyclin CDK complexes को inactivate करना होगा, cell will degrade these cyclins, CDK will be inactivated by itself. Okay, so cyclin levels in the cell are controlled not only by changes in cyclin destruction but also changes in cyclin synthesis. कई बार क्या होता है कि एक तो ये हो गया कि इसका destruction कर दो, इसको degrade करवा दो, दूसरा ये है कि उसके cyclin की synthesis को regulate करके, means at transcriptional level. Now we will see. Okay, so we will go to this particular PDF. So there is there is generally 2 family. One is SCF and second is CDC20. This CDC20 is actually having some activating function for this APC. This APC protein will either bind to CDC20 or CDH1. We will see how. So, okay, I will discuss this also, but before discussing this, I want to show you something. Okay, so actually we were here. So now you will understand that we have discussed CDC2, CDC25 and CDC20. Okay, now you recall that CDC2 was a was a kinase actually called CDK1. CDC25 was a phosphatase actually having opposite function of CDC2, means it is a kinase, it is a phosphatase. But CDC20 is completely different. It is having some ligation function. So this is called E3 enzyme of ubiquitin ligase that are generally E3 enzyme in this proteasomal mediated degradation. Okay, we will see, but you need to remember that these three are different, not the same, as the name is suggesting they are same. They are not the same family. Now I want you to focus on these two diagrams. One is this, that is control of proteolysis by APC complex, anaphase promoting complex. Okay, and the second is control of proteolysis by this SCF, which I told you that it is a combination of SKP1 cullin protein and F box protein. So this is F box protein, and the other two proteins are here. One is your SKP, and the blue color protein is your cullin protein. So okay, we will discuss this, but before this we need to discuss this particular. Now, the first regulation of proteolysis or ubiquitin-mediated proteasomal degradation occurs through this particular APC or anaphase promoting complex. So this is your APC. Okay. Oh, sorry. This is your APC, and this is some activating subunit which can be either CDC20 or CDH1. That is CDC homolog 1. Okay, CDC20 homolog 1 protein. So या तो ये एक्टिवेटिंग सब यूनिट जब इसके साथ बाइंड करेगी, so it will activate your APC protein. So it will be activated. Once it is activated, because we know this APC is a E3 ubiquitin ligase, E3, and this E3 ubiquitin ligase is required for the ligation of ubiquitin polymers, sorry, ubiquitin monomers to the target proteins. Now see, this is your CDK cyclin complex. So suppose this is CDK1, this is M cyclin. The M cyclin, you know, this is called cyclin B. So this cyclin B of M phase cyclin should be degraded so that CDK can be inactivated. So you need to ubiquitinylate this particular cyclin or cyclin B so that it can be degraded. So what will happen? These two enzymes, one is this E3, E1 and E2. So E1, E2, E3, all the three will be required so that this particular protein or target cyclin B can be ubiquitinated. This is called polyubiquitin tag. Once this tag is put over some target protein, it will undergo degradation by proteasome. This proteasome is 26S proteasome. I hope you will get a simple understanding of this particular thing, what is going to happen, what is happening here. Similarly, when this SCF or this SKP1 cullin and F box protein making a complex, okay, so this particular complex is getting activated and will bind to phosphorylated targets. So suppose there is an inhibitor or P27, we have discussed this P27 in retinoblastoma portion. So this P27 is phosphorylated by CDK2. Once this is phosphorylated, it will have phosphate over it. Once it is having phosphate over it, it will be recognized by F box protein of SCF, F for F box protein. So it will recognize this particular phosphate, and because it is a E3 ligase of ubiquitin mediated proteasomal degradation, this is E3, so it will be required for proteasomal degradation. So two more proteins which will be required are E1 and E2, and finally this E3. I will tell you what are the function of these E1, E2 and E3. For now, simply recognize it as E3. We will need E1 and E2 also, so that this particular P27 or CDK inhibitor protein can be phosphorylated and degraded. Once the CDK inhibitor protein is degraded, so that the cell can move out of some repression, so that it can move from one phase of cell cycle into the next. Now we will see what are these three enzymes, E1, E2, E3. I hope some of you have read about this proteasomal degradation. Some of you may not be able to get this who are in their BSc standards. So okay, so there is no problem. We are going to discuss this particular process, actually how this proteasomal degradation is going to take place. This particular diagram is from Leninger. I'm having 6th edition and page number 1147, chapter 27, and see this ubiquitin protein. So first of all, you should know what is ubiquitin protein. So ubiquitin protein is a protein which is made up of 76 amino acid residues. The most highly conserved protein known till date, and it is having 76 amino acid residues. सभी organism में identical होती है because it is conserved protein and it is similar in yeast to humans, means in all animals it is almost similar. 76 amino acid protein, and each protein is having two terminus. One is called N terminus, one is called C terminus. So this is the C terminus of this particular ubiquitin, and generally at the C terminus there is an amino acid called glycine. So this is very important, that this particular glycine is responsible for the formation of thioester bond. Okay, so who is the enzyme? This E1 enzyme is called activating enzyme. Simply, there is no name. These are very simple terms used here. E1 activating enzyme, E2 conjugating enzyme, and E3 is a ligase which is used to ligate this particular ubiquitin to the target protein, so that it can be tagged to for proteasomal degradation, So this E1 activating enzyme, by utilizing one ATP, because this enzyme or activating enzyme is having SH group coming out of some cysteine residues. Okay, so some cysteine is having SH group, and this glycine in ubiquitin is having this C terminal COO group. Now the ATP will be consumed, and the energy of ATP will be converted into AMP and PPI. Now you will see that one ATP is degraded, but energy of 2 ATP will be utilized because the ATP is directly converted into AMP. So remember, for each bond, means for each bond of this particular, okay, so formation of this thioester bond require 2 ATP molecules. Okay, so once this E1 activating enzyme is forming a thioester bond, this is thioester bond which is between the glycine CO and SH of cysteine of E1 enzyme. Function of E1 is over. This activating enzyme is itself got attached with ubiquitin protein. Okay. Now E2 enzyme will come, which is a kind of conjugating enzyme, and it will replace the E1. Now see, okay, so this E2 will come. It will also have cysteine group, which is having a thiol or SH side group or R group. Now it will replace your E1 along with its SH group, and itself will bind to the glycine, C-terminal glycine of ubiquitin. In the third step, this ligation reaction will take place, and the question has been asked several times: what type of bond will be formed, what type of amino acid will be linked in the target protein, and like this. Okay, so I will answer those questions in this particular text here. This E3 ligase is required so that your E2SH can be freed, and your target protein is having some lysine residue. So this lysine residue is having an amino R group. This is not this particular amino group, because this is not free. This is forming the peptide bond protein. Okay, but the R group of lysine is free. So if this is alpha carbon, this will be beta, gamma, delta, epsilon. So epsilon amino group of lysine will form a bond with your ubiquitin. So here this ubiquitin glycine C double bond O is forming this particular epsilon amino group is forming a bond with lysine of target protein, so that this ubiquitin protein can be tagged over this particular target protein. So now what is the important point here to note is this particular amino group is not from the alpha carbon. So it will not called the peptide bond or amide bond. This is called isopeptide bond because this is amino group, but not in the not over alpha carbon. It is over the epsilon carbon. So I would like to annotate here. This is alpha, this is beta, this is gamma, this is delta, and this is epsilon. So this particular epsilon amino group will form a bond with your target with your ubiquitin, glycine. So here it is glycine, here it is lysine. Lysine is K, glycine is, so simple trick is GK. So the G will form a bond with K, but this bond cannot be called peptide bond, but this bond is called isopeptide bond because I have told you already. Okay, so this is from the epsilon carbon. I hope this is clear, and if you're having query, I'm looking into your queries also at the same time. Trishla, so CD is not responsible for cyclin regulation, right? Okay. Trishla, actually, CDK1 is itself got inactivated. Yes, some text is saying that it will help or it will trigger the cyclin degradation, but it is not clear. Actually, this is not given anywhere. I did not find it. If you're able to find it, please let me know. I will try to find it. Okay. So actually, it is not given anywhere. It is only written in some books. Actual mechanism is this, how the cyclin is getting degraded. Okay, fine. I hope this complete mechanism of ubiquitin mediated proteasomal degradation is clear, and this GK bond is formed, that is called isopeptide bond, and you will remember this particular thing, then you will not going to make a mistake if somewhere you will face this particular question. So you can read this text, but okay, so some proteins within cell are having only little life, that is 30 seconds in eukaryotes, but some proteins are having up to 110 days in case of hemoglobin, for example. Your hemoglobin will be formed when your erythrocytes or RBCs are forming, and it will be there until and unless your erythrocytes or RBCs are destructed in your spleen. So spleen is known as the graveyard of RBCs. So this hemoglobin is having the highest, means this particular lifespan of 110 days. Then the defective proteins and those with characteristically short half-life are generally degraded in both bacterial and eukaryotic cells by the selective ATP-dependent cytosolic systems. We know these type of systems are called ubiquitin-mediated proteasomal degradation kind of things. But second things also have reported in vertebrates, that is in lysosome. You know, lysosomes are having cellular debris, and this particular cellular debris are also including proteins, and these proteins can be degraded by the acid hydrolyses, which are enzymes present within lysosomes, which were acting over low pH. Okay, so this type of things can happen in vertebrates also. And E. coli, this particular ATP dependent protease is called LON actually. Okay, so this LON is for long form of proteins, which observed only when this protease is absent. If this protease or LON is absent, the long forms of protein can be seen because protein is no more degraded. So you can observe the long forms of protein in its absence. Therefore, this particular proteasome is termed as LON in E. coli. 2 ATP molecules are hydrolyzed for every peptide bond cleavage. So ubiquitin, I have told you already about this. Now, this is important. One protein, this is a target protein. Okay. Oh, sorry. This is, okay. So this is a target protein and it is having some polymers. This is called polyubiquitin. So this is the target substrate protein, and this is polyubiquitin polymer attached over this particular target protein. Now, this will act as a tag or flag, which can be recognized by your system. This is called proteasome. this proteasome is called 26S proteasome. So this complete structure is 26S proteasome. This proteasome is made up of two subunits, actually three subunits. One is 20S core subunit, which is having alpha, 2 beta, and here is alpha. Alpha, beta, beta, alpha. So there's 2 alpha, 2 beta, but the 2 beta in between. So this is forming the core, and this is large structure. So this is having more weight. So 20S, whereas the two regulatory particles are both side of this core particle. These are called 90S regulatory particles, 90S regulatory particles on both ends of this core. So act as a flap or cap over this particular proteasome, and this is hollow structure in which the protein is going to degrade by this core particle. Okay, so the complete structure, which will include two 19S subunits and which are regulatory subunits and 20S core particle, is known as 20S 26S proteasome. I'm clearing all this. Okay, fine. So 6 of the subunits are ATPases. इनमें से जो core particle के बीटा सबयूनिट्स होते हैं उनमें से 6 सबयूनिट्स ATPase का फंक्शन करते हैं, ATP को ब्रेक डाउन करके अपनी एनर्जी ऑब्टेन करते हैं. प्रोबेबली फंक्शन इन अनफोल्डिंग ऑफ यूबिक्यूटिन प्रोटीन्स. ये ATP कहाँ पर रिक्वायर्ड होती है? सो दैट द यूबिक्यूटिनेटेड प्रोटीन्स, यानी कि जो टारगेट प्रोटीन्स हैं उन्हें अनफोल्ड किया जा सके, and translocating the unfolded protein into the core particle for degradation. So 6 of the subunit in this particular complex. Okay, so going to do ATPase, means ATP hydrolysis, which function to unfold and translocating this target protein to the core particle. And then 19S particle also have this. Okay, so this 19S particle, which is having some regulatory role, also you de-ubiquitinate the protein as they are degraded into proteasome. So remove the ubiquitin tags over the target proteins, so when they are undergoing this degradation. I hope you are able to get the thing. Actually, I want to tell you these three enzymes, E1, E2, and E3. This is E3 actually, APC in this case, and in this case SCF. Okay, so these are E3 enzymes or ligases which are responsible for formation of glycine bond with lysine, and these type of bonds are this type of bond, because this is the target protein, so this is isopeptide bond. So this target protein lysine and ubiquitin is linked via its glycine. So GK bond is formed always. Okay, then we are moving to our next discussion. Once this APC or the SCF complex is adding the ubiquitin tags over this Securin saccharase complex, this Securin is degraded. Okay, so this is an inactive state because Securin is forming a complex with saccharase and hiding its catalytic site. When the ubiquitin tag is added over Securin, the Securin will undergo some proteasomal degradation. This is kind of recycle bin, and this is broken down. Once the Securin is broken down, your saccharase catalytic site is free to act, and it will degrade your cohesin proteins or cohesin SCC1 protein, so that the ring can be opened and these two sister chromatids are separated, means they are separated, they can move towards the opposite poles. Students, if you are facing any query or any problem up to here, you may ask me. I will explain again. Okay. So otherwise, we are now we need to read it from Pathfinder, and whenever required, I will finish this particular topic with the help of Cooper. I'm giving you 2-minute break so that you can analyze things what we have discussed up to here. Okay, fine. Then most of you have studied it earlier also, and those who are studying it for the first time, I request them, please ask your queries, or otherwise we are moving to our next portion. Okay, so now we have discussed all these things. When you read the text, you will get the things, some terms. For example, RBX1 is given here. We have not discussed. Okay, I'm telling you other things we have discussed. So what is this RBX1? So we know we have discussed the two proteins. One is this SCF, and the second is this APC. Okay, so this is full form for anaphase promoting complex cyclosome. These are responsible for ubiquitination and destruction of S and M phase cyclins. Now, remember, this particular complex is required to remove the destruction or remove the S and M phase cyclins. This SCF complex is required to remove the G1 phase cyclins, actually. Now, the complex also contains a ring domain protein. This SCF protein is also having a ring domain protein that is called RBX1 protein. This is not given in the book or diagram I have discussed with you all, but this SCF protein is also having one protein, either this or this, which is called as RBX1. Okay, so the another protein which is having the same function of E3 ubiquitin ligase is this particular protein APCC, responsible for ubiquitination degradation. Is this. So now, but unlike SCF, yes, this protein does not have a F box protein. Okay, we have seen already in the diagram. Now we have discussed all these things yesterday. Okay, so this particular diagram, you may face problem. I'm explaining this to you. So the A is showing the wild type when the V1 and CDC25. This is positive. So it is having a kinase which will phosphorylate your inhibitory positions, and CDC25 is going to remove all those phosphates so that the cell can easily move from G2 to M. And once the M phase is over, the cell will undergo karyokinesis and cytokinesis to make two equal cells out of this big cell. Okay. So the cell size is increasing in G2, but in M, the cell will be divided in between and the two equal cells will be formed. If somehow one is mutant, if the V1 is negative, so the kinase will be absent. It will not going to phosphorylate your inhibitory positions, that is generally tyrosine 15 or pyridine 14, and you are having this phosphatase. So there is no requirement of this phosphatase if you do not have this particular kinase to phosphorylate those inhibitory positions. So in this case, your cell will not grow in size. Okay, and this type of mutants will called V1 mutants, or the V1 is absent because the cells which will form after the cytokinesis of this type of cells or this type of mutants will be smaller in size than this one. So actually this type of cells, V shows the effect of loss of function, mutation of V1 gene. So this is negative, this is not present. So kinase is absent, so the cell divide prematurely. So the cell will not grow in size and divide before maturation, forming smaller cells. So small is a term in Scottish, this is known as V. So therefore this term was given V. I told you yesterday also. Now suppose if you do not have this phosphatase and if you are having kinase, what will happen? So kinase will phosphorylate the inhibitory position so that the phosphatase is no more available to remove those particular phosphates. Your cell will not switch from G2 to M phase and continue to grow. सेल की ग्रोथ होती रहेगी एंड सेल जो है G2 से M phase में ट्रांसफर या ट्रांजिशन नहीं हो पाएगा. ठीक है. So this is very clear from this particular diagram. We also have discussed this particular thing that there is V1 inhibitory kinase, and we have some activating kinase CAK, which phosphorylate tyrosine at 15th position and 161 position phosphate, threonine. Now CDC25 AOC is removing this particular inhibitory phosphate at 15th number of tyrosine, so that this particular complex of CDK1 and mitotic cyclin B can be activated and cell can switch from G2 to M phase. This we have discussed. Then I also told you some of the inhibitors of which are in two families, INK4 family and CIP-KIP family. If you read these things, you will came to know that we have discussed all these things in previous class. Okay, this MCM protein or MCM helicase also we have discussed. This is made up of 6 subunits. MCM1 is not there, but 2 to 7 or 6 related polypeptides are making this MCM hexamer. So MCM4 mini chromosomal maintenance, and this particular MCM protein is forming a ring-like structure, pre-replicative complex, and this pre-replicative complex is having two different. One is called in early G1 CDC6 and CDT1 associated with the ORC, this origin recognition complex, along with the MCM protein. They are all making the pre-replicative complex, and that is the initiation site for replication in S phase of cell cycle. Then we have discussed these things also. Everything is discussed. Okay, now. There are some terms, amphitalic, monotalic. Okay, so you will came to know everything is written in text, and if you're having this particular book, this is not a problem that you will discuss these things. You will able to understand at least. This is not very complex. Okay, so I'm moving to directly over this particular portion where this spindle assembly checkpoint SAC is required. So this spindle assembly checkpoint is to check whether your chromosomes are properly attached over the metaphase plate so that they can equally distributed between the daughter cells. So this SAC is composed of several proteins that are conserved from yeast mammals. The core component are made up of two proteins. One is called MAD1, that is mitotic arrest deficient protein. MAD2, MAD3, there is three related forms. And the second is, instead of these, actually BUB-R1 protein is present in mammals, and 1 and 3 is present in, okay, so one thing is this and this. Okay, so little complex from this particular text. We will move to our reference book so that we can better understand this, what is happening here. Okay, so this is also you will find in your pathfinder that this gamma tubulin and other proteins are required for the formation of spindle formation. The centrosome maturation and spindle assembly also involve the protein kinases, which are under two families. One is called Aurora and Polo-like kinase families. So located at the centrosome only. What is centrosome? Centrosome is centrosome. Okay, so one thing you need to know. Okay, so here, when the cell is in interphase, so this is centrosome, which is made up of a pair of centrioles. So before cell enter into the M phase, okay, so interphase only, see, the cell is in interphase, but you will see here it is only one centrosome, here it is two centrosomes. So centrosome divide in interphase only before entering the M phase, so that each daughter cell will receive one centrosome. So the centrosome is made up of one pair of centrioles, and the microtubules are coming out of this particular complex. And for the spindle assembly and all these things, you will require an additional family of protein kinases called Aurora and Polo-like protein families. Okay. Now we were discussing spindle assembly checkpoint and progression to anaphase, how this is happening actually. So based upon how your chromosomes attach to spindle, so this is centrosome, and they are forming the poles within cell. Okay, so okay, before discussing this, I would also like to let you know that this nuclear envelope has been disappeared from this particular cell. There is no nucleus. But in some cells, for example, in yeast cell, there is a nucleus, so that the nuclear envelope will not disappear. Okay, so this type of mitosis is called closed mitosis, whereas the open mitosis is when the cell nuclear envelope has been disappeared, and in cytoplasm there is two poles are formed by these centrosomes. Okay, if the two poles are formed within the nucleus itself, then this type of mitosis is called as closed mitosis. Generally, this type of mitosis will result into unequal cell size. Daughter cells will not have equal sizes, whereas open mitosis will always result into the equal cell formation. So the daughter cells will be similar in most of the cases. And so I had discussed this yesterday also, that this lamin proteins or nuclear lamina will be formed by lamin proteins, and these are like bladder of your football. So if you suppose nucleus as football, so some structural maintenance from inside support is from the nuclear lamina, and this nuclear lamina is made up of nuclear laminin protein or filaments. So these filaments are phosphorylated by this particular kinase CDK1 when it is active and making a complex with cyclin B. So this is called MPF, maturation promotion factor. So these are phosphorylated, and these polymers or dimers then segregate. Once they are segregated, they will move to the endoplasmic reticulum. Okay, several evidences showing that these will move to the endoplasmic reticulum, and once they're moving to endoplasmic reticulum, the nucleus will disappear. Once the kinases and this cytokinesis or karyokinesis is over, endoplasmic reticulum again release these polymers, again release these dimers. They will making the nuclear envelope again once the mitosis phase is over or towards the end of telophase. Now, most importantly, we need to switch to SCF, and I was here actually. So based upon how your microtubules, suppose these are positive ends coming out of your poles, okay, so there is a possibility that these ends are free. So these are called astral microtubules because they're forming astral-like structures. If your microtubules are coming over one another, then this positive pole of one microtubule is overlapping with positive pole of another microtubule. So this type of microtubule then are called polar microtubules because the two poles are interacting with each other. There is two more possibilities, that the kinetochores of these chromosomes, which are structures where your sister chromatids are joined, if the kinetochores are attached to spindle apparatus, then these are called kinetochore microtubules. Whereas if your spindle is attached to chromosome other side than this kinetochore, then these are called chromosomal microtubules. So four types are possible based upon the arrangement of chromosome in your. Now, the progression from metaphase to anaphase results from the ubiquitin mediated proteolysis of key regulatory proteins triggered by the activation of, okay, so see here, your cohesin protein, when the cell is in M phase and in metaphase stage of cell cycle. So actually before your securin is degraded and this cohesin is being removed from in between these two chromosomes, sister chromatids, Many things are happening actually. So this spindle assembly checkpoint is to check the progression to anaphase is mediated by this activation of anaphase protein complex APC, ubiquitin ligase, that is APCC. Okay. So unattachment kinetochores lead to the assembly of complex of if the spindle apparatus is not attached somehow to this particular kinetochore, okay, of this chromosome. So the two protein, one is called BUB, and one is called MAD, will bind. Once they bind, this MAD protein will be freed from this complex and it will inhibit the CDC20. Once the CDC20 is inhibited, CDC20 is no more available to activate the APC. Now, from our last discussion, you can see from here that this APC is needing this activating subunit called CDC20 or CDH1. If this is not available, this will not be activated, and this will not be activated, it will not be ubiquitinated. So it will not be degraded. So this complex will remain there. That is called metaphase promoting complex, MPF. So now again, so second case is when all chromosomes are aligned on spindle. So kinetochore is already attached to many of the spindle fibers. So there is no free kinetochore. And in this case, this CDC20, instead of binding to this MAD protein, will bind to its APC partner. And this APC, when binding to this activating subunit, it will become activating. So this is having regulatory role, this is having some catalytic role, this will be activated. Once this will be activated, this will do two type of proteolysis. One, it will degrade the cyclin B by ubiquitin mediated proteasomal degradation. This is ubiquitin, the polymer will grow, and ultimately the 26 proteasome will come and will degrade this particular cyclin B so that the CDK1 can be inactivated. Second thing is, this will degrade your Securin out of this Securin saprase complex so that this saprase complex can be activated and will remove your cohesin in between these two sister chromatids so that this can move, this particular chromosome or sister chromatid can move towards the opposite poles of cells so that equal number of chromosomes can be distributed to two daughter cells. Hope this type of regulation is clear. This is called anaphase promoting complex. Okay, so APC. If you're having a doubt from this portion, you may ask. Okay. So once the cell is moved from metaphase to anaphase, once your sister chromatids are separated, what the anaphase will followed by telophase, and the cell will completing its karyokinesis followed by cytokinesis. The cytokinesis is in mammalian cells or in animal cells is mediated by a contractile ring of actin and myosin 2. Please underline this. This is made up of actin and myosin 2, whereas in plants, this, okay, so in plants, in higher plants actually, rather than being pinched off half in between the contractile ring, there is no contractile ring in plants, but they are divided by some other mechanism. They are following cytokinesis. Okay, so this is simply the animal cell will divide by contractile ring. So it will pinch off in between. So the two cells will form, two daughter cells of equal size, generally equal size, or sometimes unequal size will form in simple organisms. Okay, so in case of plants, in higher plants, these Golgi vesicles will start accumulating at metaphase plate and they will release some early cell plate material. So early cell plate will be formed. In case of higher plant, the cell plate will lead to the synthesis of cell wall. So the material will be deposited and thus this will extend towards the periphery or the plasma membrane. Ultimately, some of the junctions will form. These are called plasmodesmata in higher plants. So the new cell wall will be formed with some plasmodesmata and two daughter cell will result. You also know that beside mitosis, which we have discussed as M phase of cell cycle, there are meiosis also. Meiosis is required to make some of the gametes. Okay, so these gametes are haploid in nature, and to restore the diploid version of an organism, the fertilization is required. So in contrast to mitosis, meiosis results in the division of diploid parental cell into the haploid progeny. The N haploids will be formed. Okay, so this can be divided into two versions. One is called meiosis 1 and meiosis 2. What is the major difference? Meiosis 2 is very much similar to our mitosis, whereas meiosis 1 is different because it is having so it is having a prolonged prophase actually. So the prophase of meiosis 1 is much more complex than mitosis. So meiosis 2, so you will see here that the diploid cell, when need to make gametes or haploid cells, okay, so need to undergo meiosis and then meiosis 1 and then meiosis 2. Under meiosis 1, it will undergo prophase, and prophase is made up of 5 stages, which are given very clear in this particular book: lactotine, zygotine, pachytine, diplotin, and dikinesis. Now, you need to remember certain things about these stages of cell cycle. Meiosis 1. So during lactotine, recombination frequency will increase. So the recombination occur at a high frequency during this particular meiosis, complete meiosis, but starting with lactotine, so is initiated by double strand breaks that are induced early in meiotic prophase, that is lactotine. So in lactotine, underline this, you will see certain double strand breaks in your DNA. Okay, so the double strand breaks are seen in lactotine, followed by zygotine. In zygotine, you will form the sign of SC. So the short, okay, I will tell you a trick to remember this. Actually, this is a bit complex. So during lactotine, you will see double strand breaks. Okay, so these breaks, double strand breaks, will be visible during your lactotine. Then you are undergoing zygotine. Zygotine is characterized by SC. So this synaptonemal complex, or simply the synapses, will occur during this particular phase of cell cycle. So the synapses or synaptonemal complex will be formed during the second stage of your prophase 1 or meiosis 1. Then pachytine. Pachytine is characterized by formation of chiasma or chiasmata, cross-like structures. Sorry, it is very much chiasma, which are something like this. These type of structures are visible in your pachytine stage of cell cycle. Then diplotin. Diplotin is characterized by bivalent formation. So if the question is like the bivalents are formed under which stage of cell cycle? So always under diplotin, bivalents are formed. I will tell you in detail what are bivalents, but before that I would like to finish it like this. Then ultimately it will undergo dikinesis, and you will see it from here. If you have recorded all this, then we are moving to our next page. So very clear here. This lactotine is something like this. In pachytine, you will see these chiasma-like structures, X-like structures, which are forming bivalent chromosomes in diplotin stage. So these are bivalent chromosomes. If not visible from here, you may. So chiasma, the crossing overs. Okay, chiasmata for plural, chiasma for singular. Synaptonemal complex disappear at the diplotin stage, and the homologous chromosomes separate along their length. Importantly, how they remain associated with these structures, which is critical for the correct alignment at the metaphase plate. So this stage, each chromosome is paired. This is paired chromosomes are called bivalent. So these bivalent chromosomes are characteristics of diplotin. Also, one more important thing I would like to tell you here, this diplotin is the cell stage where human, not only human, the oocytes or eggs of these animals are trapped. So the first trap is during this diplotin stage. There's two type of traps during the maturation of oocytes. The first trap is under this diplotin, and the second is, I will tell you in text. We are coming to that point. So at this stage, this dikinesis, the final stage of prophase 1 is transition to metaphase during which the chromosome become fully condensed. So here you can see fully condensed chromosomes. So this is characteristic of dikinesis. Then this synaptonemal complex is something like this. There is chromatin or the DNA, and there is lateral element. Okay, so this is DNA. This is lateral element is forming zip-like structures. So this particular complex is called synaptonemal complex, which is visual during zygotine stage of cell cycle. Then you are having these type of structures are called bivalent chromosomes. These are characteristic of diplotin stage. So eggs or the oocytes are under this diplotin stage of cell cycle. This is the first check. Okay. Okay, so fine. So it is given actually here a regulation of oocyte meiosis. We are running out of time, so I want to finish all this, and actually everything is written in this particular PDF. Hope you all are having this PDF. So I'm doing hurry. Now, vertebrate oocytes, or these are called eggs, are particularly useful models for the research of cell cycle in part because they are large size. They can be easily studied. They're having large size, and this size is up to 100 micron. Okay. So even the frog oocytes are even larger. They are having up to 1 mm size. Okay, so these are having large size, so therefore they can be manipulated within the laboratories. Okay, so one is maturation promotion factor. Actually, was discovered in frog oocytes only, and this is a combination of a complex of CDK1 with cyclin B from frog oocytes. Now, what is the question is how this regulatory points or the checks are or these development or maturation is being regulated. First regulatory point in oocyte meiosis is the diplotin stage. The question has been asked many times, and I have earlier also told you that this diplotin stage is very important and is characterized by formation of bivalent chromosomes. So you will see bivalents during this particular stage. Okay, so after diplotin, oocytes will remain arrested in this particular phase, in this diplotin stage of meiosis 1, up to 50 years in humans. During this particular diplotin arrest, this is called diplotin arrest, the oocyte chromosome decondenses and are actively transcribed. यहाँ पे जो होगा mRNA का synthesis होता रहेगा, actively transcribed. So therefore, there is a tremendous growth in this oocytes. Human oocytes, for example, are about 100 micron in diameter, therefore, or up to 1 mm in case of frog oocytes. So it is all told. Then the second arrest. So this is metaphase 1. Okay, so the second arrest. Oocytes of different species vary as when the meiosis resume and the fertilization take place. So here. So the first arrest is called in diplotin stage. So this diplotin arrest is first arrest, and then oocyte growth will take place during this particular arrest because the mRNA is actively formed. So the size of egg will increase. Now, what actually happens in case of humans, in other species, this is not the case. In other species, actually fertilization is required to come out of this particular arrest. But in humans, this hormonal stimulation with the help of some progesterone hormone, okay, so it will relieve this particular arrest, and the cell or the egg cells or oocyte will come out of this particular arrest. So complete the meiosis 1. Then the meiosis 2 will start, and the oocytes will again arrested in some phase of cell cycle. The second phase of cell cycle is actually the metaphase 2 arrest. So again, second time, the cell will be arrested during metaphase 2. So first is diplotin stage of meiosis 1, second is metaphase 2 of meiosis 2. Okay. So then one polar body will be formed, and this particular big cell, it is divided here, but in metaphase 2, this arrest can only be overcome after fertilization. Okay, so once the fertilization is over, this particular egg will be converted into embryo, and this metaphase 2 arrest will be overcome. Okay, so I'm reviewing this. So first is diplotin stage, second is this metaphase second. So these two type of arrest has been seen in humans, and okay, so oocytes of different. Now I want to read this out. In some animals, oocytes remain arrested at the diplotin stage until they are fertilized, only then proceeding to complete the meiosis. However, in vertebrates, including us humans, resume meiosis in response to hormonal stimulation, progesterone, and proceed through the meiosis 1 prior to fertilization only. The cell division following meiosis 1 is asymmetric, and it will form two unequal cells. One is polar body, one is another big cell, egg cell actually. So the first polar body is formed when the cell is arrested in metaphase. The second polar body will form only after fertilization. Okay. So the cell division following meiosis 1 is asymmetric, resulting in production of small polar bodies, and then oocyte will large in size. Oocyte then proceed to enter the meiosis second without having reformed a nucleus or decondensed its chromosome. Then it will arrested in metaphase 2, where it will remain arrested until the fertilization will take place. So, okay. This diagram is showing that diplotin arrest, then the cell is again arrested during metaphase 2. But if the hormonal stimulation is given, it will come out of this diplotin arrest, and then the cytokinesis will result. Cytokinesis will lead to formation of first polar body, and the cell will be arrested in this particular phase, which can be overcome after only fertilization. Okay. So after fertilization, actually what will happen? Same thing has been, okay. So the things has been told that something has been discovered. If you took some cytoplasm from here and give a microinjection to another cell, which is in G2 phase of cell cycle, and thus the G2 cell can be come into the M phase of cell cycle. Therefore, it is called as MPF, metaphase promotion factor. And this type of arrest, so this can be also called cytostatic factors, because it is required to arrest the cell in this particular state G2. And in most recent experiments have been identified a protein serine threonine kinase called MOS, the essential component of CSF. So this CSF cytostatic factor is having one important kinase, serine threonine kinase, MOS, which is required to arrest the cell in this particular G2. Okay. Okay, so then we are having complex series of signaling pathways. This MOS is, which is a part of the CSF cytostatic factor. Okay, so it is undergoing some signaling pathway, MAC ERK pathway, and this MAC ERK pathway is again not so simple. It is represented like this here, but actually more complex pathway are given around page number 652. So I'm leading you or moving you to 652. So these all pathways we are going to discuss in our cell signaling classes, which will be started soon. And these complex pathways, including RAS, RAF, MAC, and ERK, so these will act like nuclear or cytoplasmic protein or some transcription factor, so that this kind of things can happen. So many things we have discussed in today's class, and this, okay, so All these things you can find from your Cooper only. And I hope this ends this particular topic, cell cycle. If I got some important information about this topic further, I will discuss in my next class. And for today, ending this class here. If you're having any queries, I'm unmuting you all so that you can ask your queries. And if you want to write in chat box, you can ask. Wait a minute. Okay, so I have unmuted you all. You can ask your queries one by one. There are only 6 members left. We have started the class with some 12, 13 members. So almost half, 50% has been left in the end of class. Yes. So, okay.
No queries, Krishna sir.
Okay, thank you, Trishla. So if you find any, when you start reading your Pathfinder or Cooper, you may ask in group. You are free to ask there. Okay, so you are most welcome with your queries. I hope you will revise this portion and please complete this particular chapter from this particular book, Cooper. Okay, so from our next class onwards, we will start cell signaling. I also want to start enzymes and their regulation, competitive, non-competitive, uncompetitive, and mixed regulation. So please let me know whether you are more interested in cell signaling or enzyme portion. I will start accordingly, or with the discussion with sir, I will start. Okay, no problem. So most probably tomorrow sir will have a class. Then I will come again with some new topic, either cell signaling or enzymes. Okay. So complete surprise for you if you are okay.
Sir, please continue with cell signaling.
No problem, then it is fine. I hope you will vote for cell signaling because we are in cell biology. Okay. No worries. We will start with cell signaling because it is also important, very important topic in cell. Okay. So I will start with cell signaling. So see you. Okay. Good night.
Bye, sir.